Greetings from the New York Botanical Garden. I'm Lawrence Kelly, Vice President for Science Administration and Director of Graduate Studies at NYBG. I'm happy to extend a warm welcome to returning viewers who were with us last week and to those of you who are joining us for the first time today. This is part 2 of the symposium, Here Today, Gone Forever, Plant Extinction Now and Conservation Strategies for Tomorrow. The purpose of the symposium is to shed light on the problem of plant extinction. Last week's presenters examined the scope of plant extinction globally and in North America. Today's speakers will continue on this theme and will also expand on strategies to minimize future extinctions. We're grateful to all of our speakers for addressing this grave and complex problem using data-driven scientific approaches, and we thank them for sharing the optimism of the solutions they propose. Today's speakers affirm what we talk about every day at the New York Botanical Garden, which is the essential role of plants in ecosystems and the health of the planet, the necessity of plants for human survival, and the importance of data and science to understand and conserve plant diversity. Before going any further, I would like to inform you that the symposium is being recorded and saved by the New York Botanical Garden, and it will be available for viewing in our online lecture library. Now, it is my pleasure to introduce our moderator for today's symposium, my colleague, Dr. Robert Nazi of the New York Botanical Garden. Over to you, Rob.
Thank you, Larry, and greetings, everyone, from the New York Botanical Garden. I'm Rob Nazi, the Arthur Cronquist Curator of North American Botany here at NYBG. I am very excited to join Larry and welcome you to this second part of this symposium. Shortly, we will proceed with pre-recorded presentations from today's 4 experts. Immediately following the presentations, we will have a live question and answer session with the speakers. Feel free to send questions during their presentations. At the lead of your question, please mention to whom you direct it. If you are using Zoom, please use the Q&A button. Also, if you have the same question as someone else, please give it a thumbs up so we know to prioritize the most popular questions. You are also welcome to use Zoom's chat feature to comment, but please do use the Q&A for your questions. If you are on YouTube, please use the chat feature to send your questions. Today's presenters bring diverse backgrounds, perspectives, and styles. In order to provide as much time as possible for the presenters and ensuing discussion, I will keep my introductions very brief and invite you to view the speakers' biographical information on the symposium website. At this time, I am pleased to introduce our first speaker, Dr. Nokwanda Makunga. Knox is Professor of Medicinal Plant Biology at Stellenbosch University. She will present Medicinal Plant Use in a Biodiversity Hotspot, a Balancing Act.
Thank you, everybody, for this invitation to participate in this workshop. I'm very excited to be presenting on the medicinal plant use in a biodiversity hotspot, a balancing act. South Africa is well known to have a large biodiversity that is highly endemic and unique in its character, with over 30,000 vascular plants that are found in Southern Africa, and this is about 10% of all global higher plant diversity. This region is more speciose than Brazil and Indonesia, and is equally speciose to China, Mexico, and a variety of other countries. There's been lots of news with respect to species extinctions that even make public platforms. And the drivers that are said to be responsible for those extinctions are linked to agriculture, urbanization, and invasion species. About 21 plant species will go extinct by 2030, and these numbers are said to increase. What's also quite interesting is the fact that although this might seem a little bit dire, The good news is South Africa has a very strong plant conservation strategy that is under the auspices of the South African National Biodiversity Institute, especially with specific targets that are supposed to assist in preventing plant species extinctions through the Threatened Species Program. So now I'd like for us to take a turn and look at what South Africans really look like as a nation. South Africans are culturally diverse, and this facilitates a very varied ethnopharmacology. This is quite important because the exploitation of biodiversity in South Africa is an important facet that connects the people to their ancestors. The South African traditional medicines markets are divided into two different forms, one being formal and the other being informal. The formal actually feeds the botanical drug phytopharmaceutics markets, whereas the traditional medicines market that is informal is servicing over 27 million consumers. This is quite important and significant because this particular market generates a livelihood for a wide range of different people. These would be collectors, traders, as well as the consumers of those medicinal plant products. The Cape flora, which is largely unique in its character, is actually well represented in the medicinal plant trade. I'm just going to take a few highlights here where we look at Aspalathus linearis, for example, and Cyclopia. And these particular plants are quite important because they are commercialized globally. There are also species such as Agathosma betulina, which also has been commercialized not only in South Africa, but in other parts of the world. So one of the questions that is always in our minds in my laboratory is whether medicinal plant use potentially drives plant extinctions. And we are quite focused in the Cape floristic region and the succulent Karoo because these are 2 out of 3 biodiversity hotspots that actually exist in the country. At the same time, we interact with a group of Rastafarian herbalists who refer to themselves as belonging to the sackcloth devotion. And they denounce Western norms. And the reason for this is driven by colonization and apartheid, where they pretty much say that their traditions have become extinct and their languages and traditions of medicinal plant use have been eroded. This particular group of Rastafarian herbalists belongs to a group of mixed-raced people in South Africa who somehow also have Dutch descendants, but they are part of the Aboriginal people of the Khoi and the San. So some of the questions that we are interested in are as follows. What are the most commonly used plant species in the greater Cape floristic region? This would then include both of those biomes. And do traders that are resident in these 2 different biomes have similar plant use patterns? Which species should we be focusing on in terms of conservation? And last but not least, what is the best practice in order to ensure a sustainable development of a medicinal plant trade in South Africa that is able to generate a livelihood for the Cape Rastafarian bush doctor? So our project really focused on the Cape floristic region and succulent Karoo, and we went to many different parts of this particular region, including the Western Cape and the Northern Cape. And then we utilized a variety of different plant use indices in order to try and understand how plants are actually being used by the Cape bush doctors. So some plants that were quite important are those that are positioned in quadrant 4. And these particular plant species include plants such as Sutherlandia frutescens, which had a very high use value. And several other species that I want to bring to your attention happen to be Bulbine, Latifolia, and the Tulbachia species. What was quite interesting is that mainly it is foliage that is actually used by the bush doctors, but there is some use of roots, rhizomes, tubers, and other underground parts. So some of the data that we were able to assemble from the study included information that was linked to the opinions of the group of the bush doctors with respect to what might be causing species that they cited as declining. Overharvesting came out tops at 43%, and then they also cited urban development as causing species declines. Some of the plants that they were interested in acquiring included Sutherlandia frutescens, Bulbine latifolia, and Tulbachia species, as I already mentioned. It seems that there's quite a common pattern here that Sutherlandia, Tulbachia, and the Bulbine are continuously coming up in these data sets that we actually generated. They also indicated that they would like to cultivate these particular species that they perceive as declining. However, there is a lack of capital. They do not have access to land. And because most of them have got a poor education, all of these are limiting factors that discourage the bush doctors from being active in agricultural production of the medicinal plants that they're interested in. They also expressed that they would strongly support community-based cultivation, and through the organization of the Cape Bush Doctors, they have been able to acquire pieces of land at some of the wine farms that are actually present in the region. So we then did a study that focused on Sutherlandia frutescens, because this is a multifunctional herbal product that is not only important for the informal trade, but is also commercialized for the formal trade. It is being cultivated at a very minor scale, and to increase the biomass production for the manufacturer, often wild collections are important. We first started off by trying to understand the chemistry of these plants and looking at the best chemotype which we would then take into a cultivation strategy. We utilized an LCMS-based approach and various pharmacological assays. What was very interesting about this data set is that it actually showed that the pharmacology is well matched to the site location of where the plants actually came from. In other words, plants that come from the Northern Cape in the Karoo and those that come from the Western Cape in the Cape floristic area are actually vastly different in terms of their chemistry. We were then able to establish a microplant culture as a means of trying to conserve these plants, because these plants are uprooted whole, or at times even the seed and seed pods are collected from natural populations. We used both a metabolomic analysis and a proteome analysis to try and understand what was actually happening with metabolism once these plants were grown under different environments that had different levels of phosphate and nitrogen. The data were interesting because those that were grown in lower levels of phosphate and nitrogen actually clustered in different parts of the PCA, and those that were in higher levels of nitrogen and phosphate were more similar to the controls. The metabolites that were responsible for these clusters happened to be polyamines, and various different carbohydrates were also very important, as these were upregulated by the different nitrogen stresses. So we continued the study by looking at the protein profiles, and it was very clear that the proteins that were being upregulated were related to sugar metabolism, nitrogen assimilation pathways. There were changes to proteins associated with photosynthesis, glycolysis, as well as the TCA cycle. We also picked up on several heat shock proteins and cell structural proteins, which are all very important in assisting the plant with a stress response. What was also key was the fact that the medicinal compounds, which are thought to be very important for the pharmacological activity, the solandiosides and the soyasaponins, also changed significantly. We have been continuing with this work where we have been chemically profiling quite intensely, utilizing an LC-MS strategy to try and putatively identify key chemical signatures that may change when these plants have been cultivated. Our focus is on the solandiosides, the soyasaponins, and various flavonoid compounds. So, ladies and gentlemen, I'd like to summarize what we've actually found with these two different studies. It is very clear from the ethnobotanical study that some plants are only being recorded nowadays as being utilized in the ethnopharmacopoeia of the bush doctors. And the conservation priorities for these particular species are currently still unknown. It is quite important if conservation initiatives that will assist the continued practice of the bush doctors in terms of medicinal uses, those conservation strategies really need to take into consideration socioeconomic factors that may lead to a sustainable exploitation of these natural resources that we have in South Africa. It is thus imperative to utilize multifaceted approaches in order to ensure sustainable exploitation of medicinal plant biodiversity. Last but not least, I think there's an excellent opportunity here to employ an endoma conservation approach together with an application of technology which will assist in preserving both sociocultural values as well as generating new livelihood assets for the medicinal plant user groups which we continuously interact with. With that, I'd like to then thank you all, and at the same time, thank all the different people that have been involved in this particular project. I'm ready to take questions.
Thank you very much, Knox. We're receiving a few scattered reports of poor video reception. And what I recommend is you look in your chat for the possibilities to rectify this. And we have a couple suggestions. One is to rejoin this webinar. And the other one is to try, if that doesn't fix the problem, then to try joining on YouTube. So hopefully these will work out well. Okay, the next speaker is Dr. Matthew Pace, Assistant Curator of the William and Linda Steer Herbarium of the New York Botanical Garden. Matthew will present Herbaria, Living Collections and Extinction, Documenting Loss, Providing Reservoirs for the Future.
Good morning. Thank you, Rob, for the invitation to speak today. I curate the Stereoherbarium at NYBG, one of the greatest biodiversity collections in the world, and I am extremely excited to address the fundamental role collections play in combating extinction. But first, I should address the Tyrannosaurus in the room. I love sharing the NYBG herbarium with the public, where discussions of extinct plants always elicits the same question. Could we Jurassic Park this extinct plant? As a child of the 90s, I understand the appeal of Jurassic Park. However, it's a work of science fiction. In short, no, it is not currently possible to Jurassic Park or de-extinct an extinct plant via extraction and cloning of DNA from a specimen. Even advances in CRISPR technology, which moves genes between species, is not capable of de-extincting a plant. Although herbarium specimens of extinct plants often do contain DNA, the degraded nature of this DNA, with large missing sections, is a major problem. Even if a living relative differs by only 0.1% of its genome, this could translate to thousands of genes. How many of these extinct genes need to be transferred to the living relative to resurrect an extinct species, particularly if the extinct genome is incomplete? As I stress in my public tours, perhaps the greatest potential of collections is that we can use them to better describe, understand, and conserve biodiversity, and to understand the forces that lead to extinction, ultimately providing stakeholders the tools for how to avoid extinction. Collections are critical components of the Convention on Biological Diversity's strategy for plant conservation. Botanical collections fall into three broad categories: natural history museums, which are herbaria of dried, pressed plants; germplasm collections, most often represented by seed banks that store viable seeds for extended periods of time, with a major focus on agricultural crops and their wild relatives; and living collections and botanical gardens, which grow live plants for display, educational, ex situ conservation, and research purposes. Each of these categories range from the massive, such as the Stereoherbarium here at NYBG, with 7.8 million specimens, to the highly local, such as the Tescue Pueblo Farm Seed Bank, built and run by the indigenous Pueblo people of New Mexico. Collections offer complementary and interconnected tools to combat extinction. Advances in digitization and linking collections, including the extended specimen network, will only augment their power. Another commonly asked question is, why do we collect specimens of endangered species? This question also periodically arises from within the scientific community, as some researchers ask if pictures and DNA samples are enough. Scientific collecting has never led to the extinction of a plant species. For example, only 2 scientific collections of Paphiopedilum cannei exist, whereas poachers, consisting of both local people and the international orchid community, have illegally collected thousands of individuals for sale. The pressure from poaching, combined with regional deforestation, is so great that within 1 year of its description, the authors of Paphiopedilum cannei estimate only 0.01% of the wild population persisted. Pictures and DNA can never replace the completeness of a collection. Additionally, collections are continually used in new and innovative ways that were unimaginable at the time of their collection. If we do not expand living herbarium and seed collections, including endangered species, we will sharply curtail the research possibilities of future generations. All collections types combat extinction by contributing to 4 main areas: species discovery and description, including taxonomy and systematics; species modeling, where the geographic and ecological origins of collections are used to understand how and why certain species and communities occur in particular places, and to where they might migrate as the climate changes; as permanent records of species and ecosystems, and by serving as genetic sources for the physical reintroduction of a species back into an ecosystem. Within systematics, our guiding principle is that a name is a path towards conservation. One species in which I have focused my research is Spiranthe cernua, which was considered one of the most common orchid species in North America. After using collections to reconstruct the evolutionary history of Spiranthe cernua, I discovered that what was previously considered to be a single wide-ranging species actually represented a complex of 4 cryptic species with much more restricted distributions. Now, instead of being widespread across New York State, Spiranthes cernua is limited to Long Island and the lower Hudson Valley, where it is in steep decline due to development. Clarifying the systematics of this group will also lead to the development of refined ex situ conservation strategies. For example, if Arkansas were to establish an ex situ living collection, we now know to collect Spiranthes cernua and the much rarer Spiranthes nicolasi, and that they should not be cross-pollinated so as to maintain their genetic distinction. Similarly, NYBG scientist Damon Little, working with Vietnamese partners, examined collections to better delineate critically endangered Cupressus tonkinensis, a commercially harvested conifer endemic to northern Vietnam, from the much more common and morphologically similar Cupressus flumbris. They were able to highlight small but significant differences between these species, such as needle shape, pictured at center, thus providing new collection-based tools for local foresters. It is estimated 35,000 undescribed plant species are currently held within collections. It is increasingly common for these often highly endemic undescribed species to be extinct when they are finally described, their original locations having been destroyed. This is the case with Schwartzia decidua, described by NYBG scientist Ben Torkey, from 2 herbarium collections collected 40 years earlier from a now partially cleared area of Ecuador. Similarly, Myconia phrynosomerderma was described by Lucas Major and Walter Judd in 2013 from a specimen collected in 1927. During the 86 years this species went unrecognized, its habitat in Haiti had been completely deforested, and thus this extinct species is now known from a single collection. This map represents the Amazon basin in green, with the 2019 fires highlighted in red. The gray dots represent collections housed at the NYBG herbarium from areas of forest that remain intact, whereas the yellow dots represent collections from areas that were once forest but have since been burned or cleared for agriculture. Simply put, it is now impossible to return to these yellow locations and recollect these species. These yellow specimens are our only record of this lost biodiversity. Discussions of plant extinction all too quickly descend into ruminations on loss. Yes, loss is a profound tragedy, but loss underscores the urgent necessity to expand living, seed, and herbarium collections. Collections are also used to rediscover species thought to be extinct, or to greatly expand the known distribution of a rare species. On the left is the freshwater alga Cara burtonii. By identifying previously unnamed specimens at the Wisconsin State Herbarium, NYBG scientist King Carroll was able to double the known distribution of this rare species. On the right is Echinodontium bluei, a decay fungus of the eastern white cedar. Previously known from a single New Jersey site, where it was presumed extinct, this species was recently found by citizen scientists in New Hampshire. Direct comparison of the new collections to the original collection confirmed the identification of this rare species. These examples also highlight the urgent need to collect and conserve organisms beyond charismatic megaflora, as these taxa provide critical ecosystem services, such as filtering water, like Cara burtonii, or recycling nutrients, like Echinodontium bluei. Without these small but critical organisms, ecosystems quickly deteriorate. The importance of collections to reintroductions is paramount. However, we are just beginning to examine the factors that contribute to successful reintroductions. Similarly, our understanding of ecosystem complexity is still rudimentary at best. Franklinia altamaha is perhaps the most well-known extinct in the wild plant, collected from a single lost site along the Altamaha River in Georgia. Although widely grown by botanical gardens, reintroduction efforts have failed. Hypotheses for this failure tend to focus on introduced root pathogens. But what if Franklinia is naturally ill-suited to this site in Georgia through an accident of glacial history, if it was actually trapped in the cell? One unexplored avenue is to use species modeling to find additional, more northern areas where Franklinia is perhaps better suited to forming viable wild populations. Conversely, reintroduction of Argyroxyphilum cayuense on Hawaii Island by Robert Robichaud of the University of Arizona and colleagues has succeeded, highlighting many of the collection-based factors that facilitate reintroduction. These include using many seedlings from demographically stable, genetically diverse populations. In this case, more than 10,000 seedlings produced from ex situ collections were reintroduced between 2004 and 2009, with natural germination of nearly 3,000 wild seedlings beginning in 2016. Seed banks and living collections, from Svalbard to the CITES Rescue Program at NYBG, are natural sources of reintroduced germplasm. There is also enormous interest in using seeds contained in herbaria to resurrect extinct plants and augment extant genetic diversity. So far, researchers have been able to germinate seeds from collections up to 144 years old. However, USDA permits require that all herbarium specimens be non-viable, thus placing conservation in conflict with the regulatory system. As botanists continue to explore the use of all collections types for reintroductions, we must work with permitting agencies to reduce potential for conservation-focused conflicts. Over the last few decades, seed banks have made enormous strides in collecting rare species, serving as proverbial arks of the modern era. However, there is much work to be done, particularly for tropical taxa. For example, Cyanea is a rare tropical lineage endemic to Hawaii. 21% of Cyanea are extinct, and 41% of extant Cyanea are critically endangered. Hawaiian institutions, such as the National Tropical Botanic Garden and the Lion Arboretum Seed Conservation Laboratory, have made excellent progress in creating seed banks and ex situ living collections of Cyanea, culminating in the collection and reintroduction of Cyanea kuhahiwa. However, none of the excellent Hawaiian seed banks house collections of the 14 extinct Cyanea. Herbarium specimens might seem like an appealing alternative, but after reviewing digitized herbarium collections, I found that only 26% of extinct Cyanea species have collections that contain fruit, as seen in the bottom right. Assuming these fruits are mature and still contain viable seed, there are broader ecological challenges to consider. Cyanea flowers are long curved tubes, corresponding to the curved beaks of individual species of nectarivorous Hawaiian honeycreeper birds. However, much like Cyanea, 53% of Hawaiian honeycreepers are also extinct. Assuming we can germinate herbarium seeds of extinct Cyanea, how can they form viable wild populations if their sole pollinator has also gone extinct? No species is an island, and we must leverage all collections types to conserve ecosystems rather than individual species. There is growing recognition that collections must expand to fully capture species genetic diversity. In the recent 2020 paper, Sean Hoban of the Burton Arboretum and colleagues found that of the 11 species they examined in ex situ living collections, a selection of which are seen here, most taxa were well below genetic conservation targets. Strengthening the taxonomic, geographic, and genetic diversity of living, seed, and herbarium collections will add resiliency to conservation and reintroduction efforts. Finally, I must stress that we need to collect everything, including the most common of species, while being mindful of extinction debt, which is the delayed loss of species due to ecological stresses, sometimes occurring decades after initial small declines. History has repeatedly taught us that common species, from the passenger pigeon to North American ash trees, can suddenly face extinction. I think it is important to end on an upbeat note, so I want to briefly summarize a roadmap to ensuring all collection types continue to combat extinction. We must protect currently existing collections with continued, consistent, and increased funding. We must expand collections and collect everything from everywhere with an emphasis on tropical biomes, recognizing that species are not isolated islands. Collections must characterize and reflect in situ genetic diversity. And finally, by implementing the extended specimen network and linking collections, we can leverage their combined power across living, herbarium, and seed collections to better describe, understand, and conserve biodiversity, ultimately providing us the tools with how to avoid extinction. Thank you.
And thank you, Matthew. And I'd like to remind everyone that you are welcome to submit questions during the presentations. It's wonderful that a number of questions are coming in, but feel free to send in more. And also please remember to indicate to whom you direct your question. Our next speaker is Dr. Reed Noss, President of the Florida Institute for Conservation Science. Reed will present toward a comprehensive strategy for preventing plant extinctions.
Hello, everyone. I'm Reed Noss. I'm recording this on Election Day, so if I sound a little jittery, I am. I'm going to share my screen now. Okay, so the extinction crisis is the greatest crisis of our time, even though most of the public doesn't recognize it as such. Of all global crises, extinction alone is irreversible. Today, I'll offer some suggestions for a comprehensive strategy for preventing plant extinctions. I personally do not want to see a single plant or any other species go extinct by human activity. The species in the photo here, Conradita etonia, I care a lot about. It's my neighbor. It occurs as a single site endemic just down the road from my home in Putnam County, Florida. And in fact, I prefer this species to my human neighbors, most of which have Trump signs in their yards. From an ethical standpoint, at least an ecocentric ethical standpoint, no acceptable level of human-caused extinction exists. But we know that some extinctions will occur no matter what we do. What we need to do then is prioritize to minimize the loss of species. First, we need to recognize that restricted range species are particularly extinction prone. Recognizing this, then we need to identify hotspots of endemism at multiple spatial scales, from single sites to entire ecoregions, even continents. We need to focus first on protecting those hotspots that are most immediately vulnerable to destruction. Then, to be sure, local hotspots and single site endemics in all regions are discovered and protected. We also need to identify specific proximate threats and mitigate them. And as a last resort, when survival in the wild is unlikely, we should pursue translocations and/or ex situ conservation. A basic framework for prioritizing sites or regions for protection is to score them in terms of their irreplaceability and vulnerability. This particular diagram is from a conservation assessment my colleagues and I conducted for the greater Yellowstone ecosystem a couple decades ago. If we were prioritizing ecoregions or sites in terms of their endemism, basically irreplaceability would increase with the number of endemic taxa in the region or site, going from the bottom to the top on the vertical axis. Vulnerability would increase with the urgency of threats, going from the left to the right. So major endemism hotspots that are most highly threatened would cluster in the upper right quadrant in this kind of diagram. These would be the most urgent priorities for protection. On a global scale, 36 biodiversity hotspots are currently recognized. These are biogeographic regions that meet the criteria of having at least 1,500 plants endemic to them and having suffered at least 70% habitat loss. So this is basically one application of the irreplaceability versus vulnerability concept. One bit of encouraging news is that, as Susan Harrison and I confirmed in a recent review paper in Annals of Botany, is that global hotspots of endemism are linked to stable climate refugia. They have weathered many past periods of climate change without losing a whole lot of species. They will still probably lose species if the current period of global warming is severe enough, but they probably won't lose as many species as those regions that are less climatically stable. But of course, by definition, hotspots face other threats besides climate change, usually most importantly, habitat loss. So what about smaller hotspots of endemism? Maybe single sites, micro hotspots. There's good reason to believe that at least some of these may be climatic microrefugia. They have retained their endemic species in situ through past climatic upheavals, apparently because they are out of equilibrium with the regional climate. Local site level selective pressures have outweighed regional selective pressures. I'm sure some of you have heard about and perhaps visited this particular site. It's the Ketona Dolomite Glades in Bibb County, Alabama, where 9 single site endemic plants were discovered since 1992. This is something of a success story also because much of the site is now protected as a Nature Conservancy preserve and additional land acquisition is ongoing. We need to keep discovering places like this. Hopefully we will. I'm concentrating on North America because that's where I've done most of my research. It's the continent I know best. These 2 maps show plant endemism hotspots in the 48 conterminous US states. The highest concentrations of endemics are shown in blue shades, followed by green, yellow, brown, and finally purple. And so this provides a good example also of how hotspots change a bit with change in the definition of endemic. The top map defines an endemic as restricted to no more than 50,000 square kilometers, whereas the bottom map shows range limited species restricted to less than 15,000 square kilometers. And you can see that in both maps, the major hotspots of endemism for plants are in California, parts of the Southwest, southern Texas, the southern Appalachians to some extent, and then finally Florida. And in the bottom map, you can see the real peaks in endemic species richness in greater detail, and the arrow there points to the Ketona Glades, which I just showed you in Alabama. If we zoom into Florida, using here looking at a similar database, we see 7 major plant endemism hotspots and a number of smaller hotspots. This is so-called rarity-weighted richness of restricted range taxa. All of these hotspots, I'm afraid, are threatened by Florida's unsustainably high human population growth, urban sprawl, and ex-urban development. Furthermore, coastal and insular areas are threatened by sea level rise. I'm going to come back to that topic in just a couple minutes. Hotspots are basically places that have a lot to lose, where multiple extinctions are inevitable if human activity continues to intensify. Thirst endemic hotspots are located primarily in regions with tropical, subtropical, Mediterranean, or warm temperate climates. These regions are where extinction rates are likely to be highest. But let's not forget or ignore the endemism and vulnerability to extinction of species at higher latitudes. Despite its apparently low number of extinctions so far, Canada has a surprisingly large number of national endemic taxa, 308 species of plants and animals found nowhere else but Canada. Some have very restricted distributions and high vulnerability to extinction. Every species is irreplaceable. For example, the Lake Athabasca sand dunes, which are circled here in red, hold 10 local endemic plants, including 2 composites, 1 grass, 1 pinweed, 4 willows, 1 pink, and 1 leadwort. With the high velocity of climate change occurring at this high latitude, these plants are almost certainly at high risk of extinction. As another high latitude example, I love this place, the limestone barrens of Newfoundland's Great Northern Peninsula, we find 3 endemic plants, a salix and 2 draba species. How these species fare under a much warmer climate, we're not sure, but it might not be too well. Or what if the government decides to allow more limestone quarrying in the Great Northern Peninsula? Clearly, protecting extinction-prone species in situ, in the wild, is the preferred option, but sometimes that will not be possible. In some cases, moving species begins to make a lot of sense. For example, if their current habitat is being destroyed by development, or if the climate within their current range is rapidly becoming unsuitable and due to dispersal limitations or particular dispersal barriers, they aren't able to move on their own to keep up with the shifting climate. Sea level rise is a very serious threat in coastal regions. Many coastal and insular populations of endemics will be inundated by the sea over the next few decades or so. This map shows model projections for 0.3, 0.6, and 1.5 meters of sea level rise in the lower Florida Keys. In particular, the big island in the middle is Big Pine Key. To the west of that, Little Torch Key. This is one of the major endemism hotspots in Florida, the lower Keys. By 0.6 meters, most of the natural habitat is gone. By 1.5, 1.6 meters, essentially all is gone. The red blob in the lower right of Big Pine Key, by the way, is the Winn-Dixie Shopping Plaza, wisely located to reduce flood risk, but not very good habitat for native species. So here are a couple of the endemics in question. On the left, Chamaecrista chiensis. There's only one extant population known of this species. It occurs on Big Pine Key, where I took that photo. Below that is Consolia corolicola. This is, according to the Center for Plant Conservation, this might be the most highly imperiled vascular plant species in the United States. There's only two extant natural populations remaining, one on Little Torch Key, adjacent to Big Pine Key, and another on Swan Key, just north of Key Largo. The Little Torch Key population is all male, and both populations are suffering from infestation by Cactoblactus cactorum, an invasive non-native moth. Also, both populations occur just barely above sea level for now. They won't be there for long. Attempts at reintroduction on Key Largo have not been successful, but the species is cultivated in at least 2 botanical gardens. In fact, I took this photo of Consolia at Selby Botanical Garden in Sarasota. Botanical gardens are probably the only hope to avoid total extinction for both of these species and many other local endemics. In conclusion, extinctions can be traced to specific ultimate and proximate threats. I consider the 3 main ultimate threats to be human population growth, economic growth, and anthropocentrism. We can't do much about these, at least not in the short term. We can keep working at the longer term and see how it goes. Proximate threats include the loss, fragmentation, and degradation of habitat, climate change, non-native species, pollution, and several others we could name. We can do something about these proximate threats, but it will be challenging. We need to identify clearly specific conservation actions, such as land acquisitions, control of non-native species, translocation, etc., that address specific threats faced by particular extinction-prone species. To be efficient, it does make sense to focus on hotspots of endemism, from single-site micro-hotspots, which occur in virtually every region, to large regional and global hotspots. They are all irreplaceable. And finally, of course, give the most urgent protection to hotspots most vulnerable to destruction within the near future, but don't wait until it's too late to protect other hotspots that aren't quite yet on the brink. We need to make sure that hotspots of endemism do not become hotspots of extinction. Thank you.
And thank you, Reed. Our next speaker is Dr. Barney Long, Senior Director of Species Conservation at Global Wildlife Conservation. Barney will present The Search for Lost Species.
Hi, my name is Barney Long, and I run the Species Conservation Program at Global Wildlife Conservation. Thank you very much for inviting me to speak to you all today. I'm going to talk to you about our Lost Species Program. This is a program to mobilize the world to focus on species that have not been seen by science for a while. It's supposed to inspire people across the world to care about species, especially those long forgotten species. It's supposed to instill a sense of adventure in the world about species conservation, and is supposed to make sure that we're all driving this concept of optimism around species conservation, and to really harbor this feeling that although species are declining the world over, it's in our power to find them, to save them, and to recover them. The Lost Species Programme at its heart is a crowdsourcing programme. We want the world to start to think about getting out there and exploring their garden, their neighbourhood, the wilderness areas by them, or completely on the other side of the world. Great example of this is in this photo here, when a photographer based in the US took his vacation time and travelled to Indonesia to rediscover Wallace's giant bee. He was extremely successful in his efforts, and here you can see him photographing the giant bee in the wild. GWC's focus is on crowdsourcing the whole world to get behind this effort to find all of these lost species and start conservation programs for their protection and recovery. Our focus specifically in terms of expeditions focuses on these top 25 lost species. At the time of making this presentation, there was 5 of these top 25 discovered. But just late last week, I had to add a new one to be discovered. So 6 of our top 25 have now been rediscovered and have conservation efforts underway due to these rediscoveries. So what exactly is a lost species? Well, GWC doesn't try and put too much of a scientific twist on this. We've literally just come up with a cutoff point to say a lost species is one that has not been seen in the wild for over 10 years. Currently, we have about 1,200 species on the list. And this list was put together by the IUCN Species Survival Commission specialist groups. And each year we go back to them and try and expand this list, revise it to make sure it's accurate and keep it updated. As you can see, only 20% of the species we have on the list so far are plants. This is probably a vast underestimation, probably reflected by the unfortunate dearth of specialist groups on plants and just the lack of available knowledge on these species. Going into the statistics of lost species, as you can see from this graphic, over 50% of lost species have been lost in the last 50 years. Most of the species are found in the tropics, A, where there is more diversity, and B, because there's less resources for surveys, expeditions, and just general lack of knowledge on the species in these areas compared to the more developed, richer nations where more studies have been conducted. Although the vast majority of lost species are thought to be possibly extinct on the IUCN Red List, it does not mean to say they all are. In fact, some are still considered near threatened or least concern. Just want to step back and give a little bit of an example, and apologies for using a mammal example here, but this is a species that I have worked on, so I thought it was a good example. The silverback chevrotain has been known to live in the forests of southern Vietnam for decades, although that was about all that was known, and it had not been seen for many, many decades. In fact, no one had ever seen one in the wild. So about 3 years ago, GWC, working with our partners, the Southern Institute of Ecology, started to look for this species through interviews and camera trapping. We were lucky enough to find and confirm the species in the wild, as you can see from this camera trap picture. But since then, we've continued those surveys and discovered a second population. We've also been able to set up anti-poaching operations and protection and conservation measures for one of the populations. We're starting to look at this second population that has just been recently rediscovered. And we are working on building awareness and conservation initiatives across the species range, including further searches to find new populations. So the search for lost species is not just about trying to find the species, it's about trying to catalyse conservation for these species once discovered and make sure they're not forgotten any longer, but actually put on the road to recovery through intensive conservation efforts. But as I say, the vast majority of species on this list under our programme, The Lost Species, is to look for species, is to encourage other people to look for species and not for GWC itself to go out and look for them. And for these groups here that you can see, we've actually partnered with either specialist groups or with conservation organisations to pull together specific lists of species that need to be rediscovered. And often we create top 10 lists to try and encourage people to go look for the most, what we consider the most important or critically lost species. As you can see, this very much falls in line with GWC's current expertise around mammals, amphibians, reptiles. But it's a way to form partnerships and really engage a conservation community around a specific set of taxonomic groups where we can go out and search for these species. We are always looking for opportunities to expand these groups, form new partnerships with other organisations, and expand the search for lost species across the world. So in summary, I would just say that the search for lost species is really about mobilising the global community, whether students, universities, local NGOs, or keen amateurs, to go out and find species that have been long forgotten by the scientific and conservation community. It's really about instilling a sense of optimism that these species still exist and they can be found, they can be conserved, and they can be recovered. Plants are hugely underrepresented in our search for lost species. And so I'm really excited to be able to talk to you today about the search for lost species and encourage you to either nominate new species for the list, to engage people to go out and go look for lost plant species, or even join us as an institutional partnership and identify a list of lost species that you would like to work with us on, and encouraging people to go try find. Please do join us and search for lost plants across the world so that we can find them, save them, and recover them. Thank you very much for your time today.
Thank you, Barney. At this time, we will have our live question, answer, and discussion with the speakers. I invite you again to submit questions using the Q&A feature of Zoom and the chat of YouTube. Please remember to direct your question to one of the speakers and feel free to give a thumbs up if someone has asked a question that you like. Now, I invite today's speakers to join us live for question, answer, and discussion. Okay, I'd like to kick things off by asking Barney, since you spoke most recently. Barney, you mentioned that the Lost Species program is one that's a crowdsourcing program. So obviously you have a lot of experience with the crowd and with marketing. And I'm wondering what your recommendations are for the best strategies for getting the word out and increasing participation and conservation of plants, especially combating extinctions. Extinction obviously is a dismal subject. So what are your recommendations for dealing with it, please?
Hi, everyone, and thanks for the questions. Well, I think everyone has different expertise in how to engage. So I think the answer to that very much depends on where you're sitting in the world. Obviously, if you're a teacher at a school or a university, then engaging with the next generation of academics and conservationists is definitely the way forward. If you're at an institution, how do you push out these messaging through your education programs or your social media accounts, etc.? To me, the most exciting parts of the search for lost species is when we engage new professionals across the world, people who have maybe just finished their degree and are looking for something exciting to get their teeth into. And I find it really enthralling to see 20, 21, 22, 23-year-olds embarking on their first expedition, their first field work, and looking for something that's potentially lost, but if they find it, it can open up not just a whole series of exciting events for them, but also potentially a complete career and lifetime service to the conservation of that species. So to me, it's always about finding those gems, those people that are going to drive conservation forward. So as much as we can outreach through whatever mechanism or method your expertise in, I would say go for it and try and find that next generation of conservationists.
Thanks, Barney. That sounds great. So Knox, this question is for you. You work with a number of groups of indigenous peoples, and I'm wondering if you find all of the people, most of the people, to be receptive to your work in conservation, or do you find some who are combative to it? And if so, how do you deal with that?
Oh yeah, that's a very interesting question. Many people whom I've been interacting with are actually very much aware that some of the plants that they harvest need to be conserved. And there have been opportunities where we've spoken to some of the conservation agencies around the Western Cape to incorporate them in projects where they're allowed to harvest at nature reserves, but at the same time, we actually monitor the impacts of those harvesting protocols. But it can be quite difficult because sometimes there's a change of hands in terms of the conservation managers, and then a particular project can actually fall away. In most instances, especially the much older Rastafarian bush doctors realize that the material that's been taken out of the wild is being taken out in tons, and that it's not necessarily always sustainable. And it's a dynamic culture, and things always are changing. And even the plants that are being collected are also continuously changing because there's trends in particular species that are heavily used at different times. But there is an awareness that conservation is important, but there's also an expression that it should not necessarily be punitive, but rather engaging with local people, because they're going to go into the reserves and collect this material irrespective of whether it's legal or not.
Thank you, Knox. Reed, this question's for you, and I'm going to read directly. Thank you, Reed, for including my boreal plant community concern in your presentation today. These biodiversity islands have a different range of risk factors on large landscapes. What might be some effective conservation approaches, particularly for remote areas, and characterizing at a plant community level?
Thank you. I think especially for a lot of these northerly or actually just high latitude, let's say, areas, obviously in most cases, the land use change is not as rapid from human development as it is at most mid and lower latitude areas. So we have a little more time perhaps to deal with problems of urbanization. Some of the really huge threats, of course, are related to climate change, because generally speaking, the high latitudes will suffer a higher degree of warming than lower latitudes. And I think all we can do is be sure, first of all, that those hotspots of endemism at high latitudes are protected, get them into some protected status, but then monitor them carefully. And if the species start winking out, I think, there are, due to climate, then really our only option is to cultivate them in botanical gardens and keep their seeds in seed banks. Otherwise, there's not really too much to do. But many of these high latitude hotspots there's probably many more that have been discovered. Some of these areas are extremely remote, so I would emphasize the need for continued botanical exploration.
Thanks, Reed. Matthew, how do photographic collections in citizen science venues like iNaturalist play a role versus herbarium collections in terms of documentation?
This is a great question, and thank you to the questioner for bringing it up. I definitely see photographic venues like iNaturalist as a real complement to physical collections. You know, one challenge that you learn when you become a botanist is that your time in the field suddenly becomes very limited, and your day is often taken up by, you know, managerial things, paperwork, other things that your job just entails. And the reality is that you can't be in the field nearly as much as you would like to. And so the community of citizen scientists who are engaged with venues like iNaturalist really do us a favor in helping to cast a wider net in terms of the plant observations that we can see. And I personally engage with iNaturalist. I upload my own photographs to iNaturalist. And I find that it's a place for dialogue with the citizen scientist community, particularly if there's a group that I focus on, for instance, Spiranthes. If I am verifying a picture that's from an unusual location that could be a range extension, you know, I'll write to the person who observed it and say, you know, thank you for posting this. You might want to contact your state herbarium or your state natural history program or natural heritage program and write to them and say, I found this, you know, could you come and look at it and you know, make a collection or verify it or just add it to their own database. So they're definitely complementary tools to help achieve the same goal.
So Matthew, since we're talking with you, I'd like to follow up with another question. So if you find a northern site that is suitable for introducing Franklinia, what is the danger of it becoming invasive and threatening native plants that are already established in the area?
Again, a really great question. And this wider issue is called assisted migration, which is the purposeful introduction of plants to locations that they were not previously known from. And that is an issue that we do have to take into account. At the same time, Franklinia is, you know, a North American native plant. You know, you could say it's even native to the coastal plain. So, you know, perhaps an issue of invasiveness, if you move it to New Jersey, might not be that much of an issue versus moving it to, you know, a physiographic region that it was never known from. And from my own perspective, conserving the biodiversity that's represented by Franklinia is a risk that I would be willing to take versus potential invasiveness. And everything that we know about Franklinia with this particular example points to the fact that it's likely not to be invasive. But of course, communities are complex issues, and we still know very little. We still know comparatively little about them. So I think that's just an argument for more research. You know, you can never know too much about communities, and we always need to find out more.
Well, Matthew, I'd like to press you a little bit on that. You mentioned that everything we know about Franklinia right now indicates it's likely not to become invasive. What do you mean by that specifically?
So we know that in botanical gardens, it's not seeding around botanical gardens. For example, we have several specimens at the New York Botanical Garden, and they're just the individual trees that were originally planted. There's never been a seedling Franklinia popping up in a part of the garden that it was never planted in. It doesn't seem to be impacting pollinator communities in the local area. It doesn't seem to be excluding other plants that it's planted around. For example, you know, some plants like black walnut actually exude chemicals from their roots into the soil that inhibit the development of other plants. As far as we know, Franklinia does not do that. Of course, you know, there's many examples of plants from Asia and Europe who are then brought to North America, where in their natural environment, they're perfectly fine and, you know, seem to play along nicely with their community members, that when they're introduced to a novel environment, take off because they're released from their ecological pressures. But again, I think because Franklinia is such a rare species and it's so rare, a lot of those issues may just not be at factor here.
Sure, thank you, Matthew. Now, Reed, you also talked about the possibility of moving plant species. So I wanted to follow up on this concept with you and get your feedback about it a little bit more specifically. And we've had a couple of questions about how to mitigate risks, unintended consequences. I'll mention a couple examples. One would be genetic mixing with close relatives in the new place into which we introduce the plant. There are other risks, of course. So I wondered if you would just comment on this. What are the best strategies for moving plant species?
Sure, very, very good question. And I did see I got a particular question for me on assisted migration. I am not a huge fan of long distance assisted migration because of the various risks that you've mentioned and others have mentioned. I would, however, keep it in the toolbox, especially if we're considering moving a species to a place that we know from fossil evidence they have existed during previous warmer periods. I think we're just, we have put so many barriers on the landscape through our human land use that species can't move like they used to. And so in some cases, we're going to need to physically transport them to where they're probably trying to get anyway. There's also some work my colleagues and I did on impacts from sea level rise in Florida. In Florida, we have in many other states with coastlines, we have many protected areas right along the coast, but then they're separated from the rest of the landscape by urban barriers and highways. And so as the sea level rises, they're unable to move across those barriers on their own. So if we can find suitable sites landward of those coastal infrastructural barriers, then I think we can safely move species without doing further harm.
Thank you, Reed. That's great. Knox, I wanted to ask you a specific question about the data that you presented on finding different chemotypes that were growing in different soils. And I'm wondering, do you have any insight onto whether those chemotypes are genetically based, or do you think this is an expression of merely ecologic differences? And the reason why this is a real question is what consequences it might have for the conservation of medicinal plants, especially if one chemotype is much more efficacious than another.
So that's an interesting question, and we've actually been working on this, and we've been looking at various other species apart from the one that I presented today, where we've looked at chemotypic differences and then also conducted genetic studies utilizing microsatellite analysis, for example. And with two of the plants that we've been working with, one of these being Aspalathus linearis or rooibos, it's very clear that these plants are not only chemically different, but they are genetically distinct. And in some instances, especially in the case of wild rooibos, you have some of these chemotypes actually occurring in very small populations in areas that are fast being turned into agricultural production land for rooibos. So there is definitely a need to continue some of this work with species such as Sutherlandia, for example, which is also in commercial production for natural products. But sometimes you have collection of the seeds and the seed pods, and that, of course, is not necessarily sustainable.
Thank you, Knox. Barney, this question is for you, I think, but I'd like all speakers to join in responding if you would like. Can anyone speak to other public or private partnerships and collaborations that are working to achieve conservation in hotspots? It seems like forging relationships outside the conservation community will be necessary to combat habitat loss. So for example, associations with industry and other practitioners that impact the land and the plants on them.
Yeah, there's multiple examples of this from across the world. I'm not sure how expansive any of them are. They tend to be relatively focused on specific issues or sites or species, but some are quite long-term and very impactful. We've heard quite a bit about the IUCN Red List today, which is the group, the classification that says whether a species is endangered, critically endangered, least concern, etc. That's actually developed quite significantly with a private-public partnership with Toyota. There's on the ground individual organizations or communities will also form public-private partnerships around specific issues. There's big global ones, like Coca-Cola works with organizations on freshwater conservation, all the way down to specific individual species approaches. Some are with extractive industries or things like hydropower companies, where the relationship is much more about social corporate responsibility and offsets. So there's all kinds of different approaches. It's a body of work that I think is increasing across the world. In some parts of the world, it's very nascent. In others, it's much more established. I think it's definitely an area where conservation needs to focus because there's a lot of opportunities there, whether it's kind of investment funding for conservation or carbon funding, that there's all kinds of opportunities there. So it's a very complicated business, still pretty new, but growing day by day.
Thanks, Barney. Matthew, are herbaria permitted to use viable seeds for germination experiments?
So as I mentioned in my talk, technically we are not allowed to. So as an institution, in order to import specimens from outside of the US, we need permits from the USDA. And the USDA is very concerned, understandably, about the importation of invasive species or pathogens. So they're very concerned about seeds of potentially invasive species somehow getting out and then spreading into the natural community and, devastating agriculture, or, accidentally bringing in an organism like citrus greening, which would, destroy the citrus crop. So all of our seeds, we say, are non-viable. And again, that's because that's what the permits require. In reality, we have not tested exactly how long our seeds might be viable in our collection. There has been some work that I know of, particularly from Europe and probably from other areas of the world, that has looked at how long, also Japan, about how long certain seeds are viable. And these were particularly looking at either extirpated or extinct in the wild species. But it is an issue that we have to deal with. And I have had requests come into the herbarium from researchers asking to germinate seeds for study. And unfortunately, I've had to decline those requests. But it is a conversation that we need to have as a natural history community with the USDA and other regulatory groups to try and work together to solve that problem.
Are you aware of anyone initiating such a conversation with USDA or other federal agencies?
I have danced around the topic with the person who I always get our permits from for the garden. But as far as I know, there isn't a community-wide effort But as you suggest, that is something that we really need to do as a community.
Okay, Matthew, here's another question for you. What is the appetite among grant providers for reintroduction endeavors, especially compared to the availability of funds for collection studies? Does it vary by region? Does it vary by other things such as perceived importance? For example, orchids have some commercial viability and are seen as flashy and popular, yet there may be other plants that are much less conspicuous but actually have a greater role in ecosystems. Or are there even other things that factor into this?
Yeah, so that's a very dense and good question. So I'd be interested to hear what other speakers have to say on the topic as well. But from my perspective, most of our funding for the herbarium comes from the National Science Foundation. And the National Science Foundation has been very supportive of collections-based institutions, both in terms of digitizing that information and making it publicly available for everyone to access, not just scientists, but school children if they want to, and also the infrastructure to support those collections, whether it's the building or the HVAC system, and this also extends to living collections as well. I honestly don't have as much familiarity with grants for reintroduction. That is a little bit outside my area. However, there definitely is public bias in terms of interest towards conservation programs or reintroduction programs that involve, flashy, charismatic megaflora. And of course, I'm an orchid systematist, so I do benefit from that. But it is a challenge. As I mentioned in my talk, algae, fungi, Rob is a sedge specialist. All these organisms are just as important in terms of supporting ecosystem services like filtering water, providing habitat, food for pollinators. So again, I'd be interested from the group, anyone who has more interest with those types of grants, what your view is.
So Reed, for example, are you able to comment on this, maybe from experience? Do you have any feedback for us about the availability of grants? I think this is really what the question is getting at, is what's the funding climate like for reintroductions, for combating extinctions, for creating preserves in a very sensible way, as you outlined in your talk?
Well, I've had some experience trying to get land protected. I can't speak to getting funding for herbaria and that kind of thing, but it really has varied so much over time with the political situation in the United States, both at the federal level and at state and local levels. Florida, for a while, had on record, actually about 3 decades, more area conserved and more money spent on conservation land acquisition than anywhere in the world, more than the entire federal government in the United States, for example. We were spending $300 to $500 million a year for land acquisition, and the sites were well prioritized using good science. That dried up completely with the change in governor about 10 years ago, and has just barely come back to minimal levels since then. So it's really, it gets down to politics, I'm afraid, of being sure that we get people in office who support land conservation, who support all these kinds of initiatives that we've been talking about here, because that ultimately determines the level of funding.
Barney or Knox, do you have anything to offer on this question?
I would just say that the global goals that are being put in place for 2030 are going to call for probably 30% of the world to be under conservation management. So we have 10 years to add probably another 10% of the world under management. Protecting land and sea is probably the most important thing we can do to prevent species extinctions and protect our home, which is our planet. With that, I hope that we'll be seeing an increase in funding for that kind of work. I think, as Reed said, it's very much a political question. So depending on what happens over the next 10 years with global geopolitics, we'll have to see whether we can hit that target of 30% of the planet protected or under conservation management. So I'm optimistic, but Land is expensive.
Thanks, Barney. And your comments followed up nicely on the topic that Stuart Pym was presenting toward the end of his presentation last week. So I recommend that presentation particularly to those who have not yet seen it. Knox, I certainly don't want to leave you out of this part of the conversation. I'm curious, how has funding been for you and your work, especially involving the intersection of conservation, pharmacology, sociology, work with indigenous peoples, on and on?
It can be difficult sometimes because the work is sometimes so diverse, so it can be difficult to source funding. But the South African government has realized that there's real value in the flora that we have, and even in the knowledge that the people have about the plants here. So there has been some concerted effort to assist with different aspects of commercialization, but at the same time being cognizant of the fact that these plants also need to be conserved. But it is difficult because drug discovery is a very expensive method or system. It also takes a really long time. So often the benefits and the beneficiation might not be immediately available to the communities where these plants are growing and who actually use them. So there can be a social conflict and even an economic conflict sometimes with regards to indigenous knowledge, medicinal plant use, and conservation. So it really is a balancing act, and it can be a tricky space to actually navigate within.
Thank you, Knox. And you certainly did a great job of presenting some of those challenges.
Rob?
Yes, go ahead.
I just want to add one thing. A lot of what we're all speaking about today is engagement. And one thing that people often ask is, what can I do as an individual? And I think one of the greatest things we can do is engage, whether that's at your local botanical garden volunteering, uploading images to citizen science websites like iNaturalist, but also engaging with your local politicians and your national politicians. Things about land conservation, funding for grants. A lot of these are national initiatives, and our representatives are not going to care about that if we don't voice our concerns about that. So if you write to your representative and tell them that making grant funding available to prevent conservation, or to prevent extinction is important to you, that tells them that is important, that they should focus on that. If a piece of land is available for sale and is going to be developed, if you voice your opposition to that, that helps potentially sway the powers that be. So again, I think engagement at all levels is something that we're all talking about and is really important.
Thank you, Matthew. I really appreciate your remarks, because I so frequently receive the question, what can I as an individual do for plant conservation? So that's very helpful. And this has been a most interesting and productive discussion. But before we conclude, I have a couple of brief announcements. We would like to continue this conversation. Plant extinction is a critically important topic. This symposium has built momentum, and the New York Botanical Garden has an enduring commitment to conservation of plants, fungi, and algae. We are considering the most effective ways to facilitate ongoing dialogue. We would like to be able to enable everyone to engage easily with others, strategize about ways to conserve plants and thus slow the pace of plant extinctions, and interact with the speakers who are eager to participate. So please watch for a follow-up survey in which we will ask for your thoughts about how best to continue this conversation. On behalf of all of us at the New York Botanical Garden, I thank My Good Fund Trust and the Andrew W. Mellon Foundation for their financial support for this symposium. Also, we are most grateful to Wes Knapp and Douglas Daly for their assistance with symposium planning. In addition, for their thoughtful contributions, I thank today's speakers, Knox Makunga, Matthew Pace, Reed Noss, and Barney Long. Thank you too for joining today's second and concluding part of Here Today, Gone Forever. We greatly appreciate your participation. If you missed any part of this symposium or wish to watch it again, the recording will be saved and shared online in the NYBG lecture library. Once published, we'll send a link to everyone who registered for today. Speakers from last week and today have presented a variety of strategies for minimizing extinctions, with recurring themes of including diverse stakeholders, forging partnerships, and planning for the long term. All speakers, Larry and I, invite you to join us in doing our utmost to successfully confront the plant extinctions crisis. Indeed, we must do our utmost. After all, we owe it to ourselves and to future generations. May you be most successful with this essential work. As always, we wish you the best of health. Goodbye, everyone.